Meiosis is a process in which one cell divides twice to create 4 unique daughter cells. These cells contain half of the parent cell's genetic information. These daughter cells are called our sex cells. In females, they're eggs, and in males, they're sperm. Meiosis is composed of 9 different stages. These are relatively similar to that of mitosis, except for a couple of key points. Meiosis can be split into 2 main stages, meiosis I and meiosis II. The first stage in meiosis I is interphase. Similarly to mitosis, during interphase, the cell will now copy its DNA, resulting in 2 identical full sets of chromatids. This cell is now diploid. In our case, the diploid number is 4 and the haploid number is 2. This means that when the cell only has 1 set of DNA, there are 2 chromosomes. When it has 2 sets of DNA, one from the mother and one from the father, there are 4 chromosomes. Within the cell, in interphase, there are 2 centrosomes, each containing a pair of centrioles. The next phase is prophase I. Similarly to mitosis, the nuclear envelope begins to dissolve, and the mitotic spindle, consisting of microtubules, extend across the cell between the centrioles. During prophase I, the chromatids condense to form chromosomes. As you know, every cell in the human body contains 2 pairs of 23 chromosomes, 1 set from your father and 1 from your mother. During prophase, the homologous chromosomes pair up. So, the chromosome from your mother pairs with the chromosome from your father. So, chromosome 1 pairs with chromosome 1, 2 and 2, and so on. What makes meiosis vastly different from mitosis is crossing over. Crossing over is the exchange of certain genes to create genetic variation. Each homologous chromosome come together in the process called synapsis to form a tetrad, and sections of the chromosomes are exchanged. You can see that after crossing over, each chromosome is identical to neither of the parent chromosomes. Instead, it created its own unique combination of the 2. Crossing over contributes to genetic variation because it takes 2 chromosomes, each expressing the same type of gene, but different alleles. For instance, eye color. But it creates variation by swapping parts and create a combination of the 2 chromosomes. So you may not necessarily have your father's eye color, instead you have your mother's. But you could then have your father's hair color and not your mother's. The next phase is metaphase I. During metaphase I, the chromosome pairs line up next to each other along the center, or the metaphase plate of the cell. The centrioles are now at opposite sides of the cell, with the meiotic spindles extending from them. Meiotic spindle fibers attach to 1 chromosome of each pair, forming a kinetochore. Next, during anaphase I, the pair of chromosomes are pulled apart by the meiotic spindle, which pulls 1 chromosome to one pole of the cell, and the other chromosome to the opposite pole. In meiosis I, the sister chromatids stay together. This differs from mitosis and meiosis II. The final phase of meiosis I is telophase I and cytokinesis. Up until this point, the cell was diploid. After the events of this phase, the 2 daughter cells will be haploid. In this phase, the chromosomes complete their move to the opposite poles of the cell. At each pole of the cell, a full set of chromosomes gather together. In this case, 2 chromosomes go to their respective sides. The nuclear membrane forms around each set of chromosomes to create 2 nuclei, and the mitotic spindle breaks down. The single cell, then pinches in the middle, through the use of a cleavage furrow in animals or a cell plate in plants, to form 2 separate daughter cells, each containing a full set of chromosomes within a nucleus. Again, in this case, 2 chromosomes. This means that each daughter cell is now haploid. This completes meiosis I. Meiosis II begins with prophase II. In prophase II, the nuclear envelope dissolves, the centrioles duplicate, and the meiotic spindle forms again. The next phase is metaphase II. In metaphase II, within each of the 2 daughter cells, the chromosomes line up along the metaphase plate. The centrioles are now at opposite poles in each of the daughter cells. Meiotic spindle fibers at each pole of the cell attach to each of the sister chromatids. The next phase is anaphase II. During this phase, the sister chromatids are pulled to opposite poles by the meiotic spindle. The separated chromatids are now individual chromosomes. Now the cell is diploid. The next and final phase is telophase 2 in cytokinesis. 4 haploid cells are formed during this phase. During this phase, the chromosomes complete their move to the opposite poles of the cell. At each pole of the cell, a full set of chromosomes gather together. A nuclear membrane forms around each set of the chromosomes to create 2 new cell nuclei. The cell goes through cytokinesis with the use of a cleavage furrow or cell plate to complete its division. Once cytokinesis is complete, there are 4 resulting cells, each with half a set of chromosomes, so they're haploid. In males, these 4 cells are all sperm cells, whereas in females, one of the cells is an egg cell, while the 3 other are polar bodies, which are small cells that do not develop into eggs.